Okay, picking up with our protein. Remember, there's 4 different levels of structure of proteins. The first one is the amino acid sequence. If it's alanine and then glycine and whatever, that's number 1. Number 2, it's either going to be one of 2 things. It's either going to be helix, it's going to be twisted around, or it's going to be pleated back and forth. Okay, that's the secondary. Okay, and this is where we left off, so I just went through that. It's not really important if you know why it's caused. It's just remember the secondary structure is one of 2 things. It's either back and forth, so it's pleated, or it's circular, that's a helix. Okay, like DNA is called a double helix. It looks like a staircase, so it's twisted like that. Okay, so that's your 2 secondary. All right, the side chains are going to extend off of the helix, excuse me. They're not involved in the bonding that creates it. So again, that's something different. But when you're looking at the secondary level of protein, is it either pleated or helix? And again, that's how the hydrogen bonds and the carbonyl groups between them, what they form. Okay, and here's a better picture of the ribbon shape or the helix. See how it's all twisted around? Okay, so this is why they call it a helix, because this is the twisting. Okay, so that's what again, this is a secondary structure, and this would be the pleated. It just goes back and forth, back and forth. Okay, so that's a secondary, either helix, spirally, or pleated. Okay, so first is amino acid sequence. Second, is it pleated or is it spiral or helix, I guess they call it. Okay, so the tertiary is when you take your 3-dimensional shape that results from that folding, and you get these attractions, so you can get your ends, your hydrophobic and your hydrophilic. Remember, likes water, doesn't like water. That's going to determine what type of environment that it'll live in. Okay, so you're going to get different ends sticking out. Oops, I'm sorry. That was kind of dumb. Some proteins, it causes what they call something called hem, which hem is like in hemoglobin. In the middle part of a hemoglobin, you have a hem, which actually is attracted to oxygen. Okay, so you can get coenzymes or cofactors. But really what happens with the third is the 3-dimensional shape. Okay, so it's going to fold, like here's helical, but is it going to really curl upon itself, or is it going to stay flat out? So the tertiary is going to be like what they call the 3-dimensional shape. After this, you're going to do a simulation, and it's going to explain these better. It'll show you better. It's hard to explain them by just, looking at you. So again, you'll see them better in that, and in that simulation. Okay. So, and then the quaternary, the 4th one, is what does it bond with? Okay. So maybe this is the primary structure is just a sequence of amino acids. What amino acids do you have, and in what order? The second, is it, helix, is it spiral, or is it pleated? Okay, in the tertiary, you see here's the pleated. It causes it to kind of like fold over onto itself. Okay, so folded single peptide or whatever. So you take this and you fold it on, and you make it more complex, I guess, is the tertiary. So they can have a beta chain or an alpha chain. Okay, and then the quaternary is when you add more proteins to it. So the quaternary is what can it add to? Here's one protein, can it add to this protein, to this protein, to this protein, and they have different chains. Okay, so the amino acid sequence, is it helix or is it pleated? I mean, how much more does it bend onto itself? How much more does it fold is your third. The folds, I guess we'd call the third. And quaternary is adding more proteins to it. So it gets more and more complex as you go through the structures. Okay. If you have a protein, the shape of a protein determines what it'll do. Okay. If you mess with either the secondary, the tertiary, or the quaternary structure of a protein, so the protein is made, if you change those, you change the shape of the protein, and it no longer can do what it's supposed to do. So proteins are very specific. That's why we have a million or so in our body. Okay, it's not like we have one protein that breaks down hamburger proteins into amino acids. We have several. Okay, so we have lots of enzymes, we have lots of proteins. Every one of them has a specific function, and it's based on their shape. So if you change their shape, they can't do what they're supposed to do. That's called denaturing, okay? The easiest way to denature a protein is to heat it, okay? Most of the time, if you denature something, and if you don't alter the primary structure, the first one, if you don't mess with those, it can be reversible. Not always, okay? But if you alter its, if you don't mess with the first, with the amino acid sequence, most of the time you can reverse these things. Not always, but denaturing basically means changing the shape or disrupting the shape of a protein, and then it can no longer do what it's supposed to do. Okay, the way you denature proteins, the easiest way, like I said, is to add heat. pH changes, really a high pH or low pH will change it. If you mechanically agitate it, in other words, if you shake it around a lot, you could break up those bonds. If you add detergents, okay, detergents will mess with the bonds. And if you add certain metals, okay, these things right here can be part of medicinal things too, drugs. They can mess with the detergent. I mean, they can mess with the function of the proteins, good or bad. Okay, it depends. Okay, so if we have a protein and we're going to denature it, so it's going to lose its shape, so it's not going to be able to do what it's supposed to do. Okay, this right here actually shows what happens when you get a disruption in salt bridges. This is what happens if somebody has cystic fibrosis. What happens is they have disruption in salt bridges, so they don't dilute the mucus. You have mucus in your lungs, you have mucus in your intestines and stuff. We dilute that with these salt bridges. And people that have cystic fibrosis, there's a change in the amino acid sequence. So they can't do this right. So that's why if you know somebody with cystic fibrosis, they have issues with a lot of mucus in their lungs. They have mucus in their digestive system. They have to take enzymes to supplement the enzymes that they don't have in their digestive system. And then they have to take treatments to get the gunk out of their lungs because their body doesn't normally do it because they don't form these salt bridges. You wouldn't think it would be that big of a deal, just one little form there, but it's huge. If you know anybody with cystic fibrosis, you know what I'm talking about. Okay, here's a way you can denature, and here's an example of denaturing a protein. Okay, we have our egg, and it has its spiral shape here, and we heat it, and now we have our new egg that's been cooked. We heat it, and now the protein isn't the same. And we can't reverse this back to the way it was. So this protein has been denatured. It no longer, now it's breakfast, but I mean, its function before was something different than it is now because we've denatured it, we changed its structure, and now it can't do what it originally was supposed to do. Okay? So denaturing a protein, if you mess with the primary, you know, the sequence of amino acids, you'll never be able to reverse it. My dogs, they're so naughty. Sometimes you can change the, it's reversible. It depends on what caused it. But again, heat, pH, mechanical agitation, all these things can change the structure of the protein. And anytime you change the structure, you change its function. They're very, very specific, and you don't want to mess with that. And anytime you do mess with that, that's called denaturing. And I think, yeah, we're going to quit right there.